Hello guys, welcome back to a new video. And in today's video we have got a very important topic related to the proteins, and I would like to include a point over there that we have been running a series of proteins, and today's the topic about the classification of proteins on the basis of amino acid sequence. But before we getting you into it, if you haven't checked out previous video or you're not clear about the basics of the protein, so do check out our videos. Let's move forward. And in today's video we have got classification of proteins. We have got classification of proteins on the basis of amino acid sequence. Like we have got different classifications. We have got classifications of a number of amino acids, and arrangement of the molecules or the atoms. We have got today we are learning the basis of the amino acid sequence. So without wasting time, let's get into it. Very first of all, I want to include a point over here that uh, simply I will be explaining you with this all topic with the help of diagram. So there are four different types, four types of uh, four types of classification, or you can say there are four types of amino acid sequence, and the first one is over here, that is primary structure, and the second one is the secondary, third one is tertiary, and the fourth one is quite. Now we will talk about each of them in deep, hopefully, and without wasting time, let's get into the primary one. So what it says in the primary structure, uh, let me do it. Okay, primary structure. Uh, you know Primary structure, as a name indicates, the primary one is a originated, or the origin, or the very simplest, the basic form of amino acid sequence, and this is the most simple form of the amino acid sequence or the polypeptide chains. Now primary structure is the one which is formed by three letter code. This is very important point. Like we have codes we have codes of uh, you know uh, we have been talking about the codes in our previous videos, like how the amino acid is arranged, and this is a sequence of the uh, the material, the information material, and this information material is in the form of three letter codes. The example is given over here. LYS is a single code, and GLY is a single code, and all between PRO, PHE, TYR, THR, all these are, you know these are codes. And the primary structure, moving forward, we've got one letter code as well, that is on the terminal ends, like K and G. Now this is kind of a different chain. Like we are just talking about uh, codes. The primary structure is the simplest one, it is the linear one, it is found in the form of codes. Now let's talk about primary structure details. So here is a beautiful example given in this picture. So you can see that this is a simple linear code. There is no that much complexity. We have got simple linear codes. This is one, you can say the code GCG. Then we have this, like we can make blocks of three. So this is how the primary structure works. I do hope that you got it. In this you're gonna see that this is the primary structure. We have got NH2 of the amino acid one end. This is the terminal 2 of the COOH, and this is the primary structure. And you're gonna see that this is linear. There is no any sort of twistation. This is all just running in a single path. Like there is no twist, or there is no any other chain present. This is a single chain, and these are amino acids. These all balls are amino acids, and they are arranged in form of codes. So I do hope that this is clear now, and let's move forward for the secondary structure. So the secondary structure is of two types: the alpha helices. Sorry, secondary structure is of two types: the alpha helices and the beta strands. We're talking about both of them. Okay, so basically, primary structure of amino acids combine to form secondary structure, and further it has got two shapes, and the one shape is the alpha helix, and the second one is beta strands, or the beta flattened sheets, or beta pleated sheets. And the example of let's move forward, then give you example of this. Okay, so here you can see the secondary structure. There is less complexity, but more than the primary one. Like you can see that here, you can see that they are in a single chain, but there are twists and more complexity than primary structure amino acid. Hopefully you're gonna understand with this diagram. Like if you compare this with the tertiary structure, of course this is less complex, but if you compare with the primary structure, it is more complex. Here you're gonna see the secondary structure, and it is of two types: the beta plated sheet and the alpha helix. Now what does mean? uh, Here you're gonna see that uh, here here they're just meeting, and they're forming bonds in line. They have got sheets formation, and one is of the twist formation, that is alpha helix, and they have got mid between bonds like over here, then here. The example of alpha helix is the keratin protein, and the example of beta plated sheet is the fibrin protein. And this is a 3D structure, and it is formed of mostly of three helices, like three polypeptide chains form a secondary structure probably. Now let's move forward. I do hope that these diagrams are wonderful and made you understand the secondary structure as well. Okay, now we have got the tertiary structure. Tertiary structure, what it means: fold helices and strands into domains. Now it is saying the same word. Like primary structure will combine to form the secondary one, and the secondary structures combine to form the tertiary structure. Uh, it is more complex. You're gonna see in the diagram, it's more complex. And 3D structure, ball-like structure. Hopefully, okay. This is. Uh, I would like to include a point over here that alpha helix or the beta plated sheet fold again and again by the ionic bonds to form the tertiary structure. And there are different bonds present. Let me show you if I'm able to show it to you, this diagram. Okay, so I'm unable to show on the diagram, but I will tell you verbally that there are ionic bonds such as hydrogen bonds between the linkages, and we have got sulfur and disulfide bonds, SS bonds in between them, that makes them stable. And yes, this is kind of about it. Like tertiary structures have got a great example, that is the enzymes. Enzymes is a very important topic, and hopefully uh, our next chapter that we'll discuss on our channel is probably the enzymes. So all enzymes are under the tertiary structure. Okay, so in this picture you're gonna see that this is a tertiary structure. This is more complex, right? More complex, more folding of secondary structure from tertiary structure. Here you're gonna see that this is more complex over here. Okay, so let's move for the quaternary structure. Biological units, functional assemblies of chain subunits. Okay, so if you talk about the quaternary structure, so they are of uh, more than one polypeptide chain. Always they have got more than one polypeptide chain, and different tertiary structure combined to form quaternary structure, you can say, in a way. And hemoglobin is a great example of the quaternary structure. Like in hemoglobin we have got two chains. One is 2 alpha helix chain, and the second one is 2 beta helix chain. Now you can see over here that the two secondary structure chains combine to form quaternary structure. This is not, we can say in a way that tertiary will always combine to form quaternary. That is not the thing. That was just a concept making thing. Like here in the case in the hemoglobin, the secondary structure, 2 alpha secondary structure and 2 beta plated secondary structure combined, and they have got uh, they made hemoglobin. So this is how it works. And okay, so this is kind of about it, and you're gonna see the diagram how complex this is. Like how complex this is. This is most complex. Over here, if you compare all those, this is more complex than all. Simply, this is simple comparison. And I do hope that you have learned something in this video. Meet you guys in the next video. Till that, goodbye.